attending our webinar today. We're excited to share with you how you can use our free image converter, MicroFile Plus, to optimize your optical microscopy data and metadata to comply with the SPARC standards for optical microscopy imaging data. I'm Macy Heel.
And I'm Aidan Sullivan.
And we're part of the SPARC Map Core team here at MBF Bioscience. If you've attended our other SPARC webinars, I'm sure by now you're familiar with us, and we're glad you're here again.
And if you missed our other SPARC webinars, be sure to check them out on the YouTube channel, sparc.science. You'll see all the SPARC webinars there that we put on by the SPARC Data Resource Center.
And for new visitors and those who are outside the SPARC program, we welcome you too. Though we'll be discussing a SPARC-specific application of MicroFile Plus today, the tool is freely available for download, and we encourage you to employ the powerful utility in your image analysis and data sharing workflows.
We'd love to hear from you and answer any questions that you may have about our presentation today. If you'd like to submit a question at any point during the webinar, just click the orange arrow button to open the GoToWebinar control panel, find the question tool panel, type the question in the box, and click send. We'll take some time at the end of the presentation to answer your questions. We also have Dr. Nate O'Connor on the line today. He's the product manager of MicroFile Plus to help answer any of those questions as well.
Today, we're going to talk about the SPARC data standards relating to optical microscopy imaging data and metadata, how MicroFile Plus is used by our team to curate data sets submitted to the SPARC portal, and how you can use MicroFile Plus yourself to prepare your data for dataset submission.
We come with exciting news today. The SPARC standards for optical microscopy image data and imaging metadata were recently approved and endorsed by the SPARC Data Standards Committee. Hey Macy, what role does an image standard play in SPARC?
Optical microscopy imaging data is a common experimental modality in scientific research and due to the variability in sample preparation, microscopy settings, methods, and devices, it can be incredibly diverse. The SPARC imaging standard ensures that a consistent set of metadata specific to microscopy imaging data is included throughout the SPARC ecosystem. It guarantees that the microscopy data hosted on the SPARC portal can be understood and repurposed despite its complex and experiment-specific nature.
That makes sense, and I could totally get on board with these standards, but Macy, can you tell me a bit more about why you think microscopy imaging metadata is important for SPARC?
Yeah, without metadata such as the histological stain or fluorescent label of the channels or the physical dimensionality of the pixel or voxel, it's really impossible for one to fully understand, analyze, or reuse an image from a given data set. By enriching these image files with essential metadata directly, we can ensure the files include that relevant and critical information about the image itself, like what we're seeing in the image and how it was acquired. Like, look at some of this gorgeous SPARC image data on the screen. These examples come from different experimental data sets and use different modalities for acquisition, channel target labels, and the size. Recording this information about the experiment within the actual file as metadata will make it easy for those in your lab and outside your lab to interpret the images on their own and as a part of the data set as a whole. If you were to download an image from the SPARC portal for repurposing in your analysis tools, you'll have all the information within the header of the file to know that, say, the fluorescent channel, green channel is chat, or it was taken from a confocal microscope, or the pixel size in X and Y is 0.5 microns. Aidan, tell us what's required to meet the SPARC standard.
So to meet the SPARC optical microscopy image data and imaging metadata standard, source microscopy image data within SPARC datasets must be converted to OMI TIFF and JPEG 2000 formats. Additionally, there is a set of required metadata that is needed to appropriately repurpose these resources. We'll review the required image metadata in detail on the next slide.
Gotcha. So how can SPARC researchers ensure that their datasets meet these requirements on the imaging standard?
So the recommended best practice is to configure your microscope software to include metadata information and use this native format whenever possible for image analysis. However, when image data is converted to another form, such as converting the Zeiss standard CZI format to the open format such as TIFF, often nearly all essential metadata is lost from the file. When this occurs as part of the experimental analysis pipeline, a final step to reincorporate the metadata is required. If this information is not part of the minimum information available, then the file is ultimately of limited value. In an effort to address this issue and make it easier for investigators to adhere to the SPARC imaging standards, we've built a freely available file converter application called MicroFile Plus. MicroFile Plus is a powerful image file converter that helps address the challenges of organizing and storing big data from modern laboratory imaging devices and retaining essential metadata. Image files from many sources can be converted into JPEG 2000 or OMI TIFF formats that allow for efficient storage, viewing, and analysis. MicroFile Plus also allows for adding and editing essential metadata to ensure SPARC image data is fair, or findable, accessible, interoperable, and reusable. MicroFile Plus writes this metadata to the header of the file, not a sidecar, and in accordance to the OMI TIFF specification. This tool makes it easy to produce image data that can be readily shared and disseminated, whether that's within your lab or to others in your department or to the public on a data portal such as the SPARC portal. We'll talk more about MicroFile Plus later, but for now let's dive into the details of what metadata is required to meet the SPARC imaging standards. This slide shows the 22 required metadata fields that are needed to appropriately interpret optical microscopy data. By writing this metadata to the image file, you ensure that your images can be understood and reused by others. Now, don't let this long list scare you. It's actually not as much work as it looks like to meet the SPARC image standards. If you take a look at the far right column here, you'll see that a lot of these fields are labeled as automatically present in the source image file. So let's break these fields down into those two categories: automatically present and present when configured at the time of data acquisition. You'll see that there are 14 fields that are automatically present, which include things like significant bits and image size in the X, Y, and Z directions. These basics are always written to the source image, so we don't need to worry about them too much. There are actually only 8 fields that may need to be manually added to the image data. These include things like physical size in X, Y, and Z, or image scaling. And again, Z would only be required for 3D image stacks. These metadata fields may already be written to your image file depending on your microscope software settings, which is why they fall into the category present when configured. Some other key metadata fields for interpreting optical image data include objective, acquisition mode, color channel name, also known as target label, channel fluorophore, and display pseudocolor. And I just want to take a second to point out that you may not have any fluorescent channels in your image, or you may have many, so the number of times you need to add these last 3 fields here will depend on that. So to meet the SPARC image standard requirements, you really just need to convert your image data to the file formats OMI TIFF and JPEG 2000 and add the 8 listed fields shown in the purple box here. But what tools are available to meet these standards? Macy's going to show you how and when you can use MicroFile Plus to make your image data fair.
Like Aiden said, MicroFile Plus is going to make it easy to ensure that the image data in your experiment meets these SPARC requirements. The application itself ingests source microscopy image data and converts these files to both OME TIFF and or JPEG 2000. These image formats were selected for the SPARC image standard because of their readable metadata storage, compatibility with open source software such as ImageJ, the longevity expected for the format support, and for JPEG 2000, the web streaming capabilities. MicroFile Plus not only allows users to view, edit, and add the metadata to their microscopy images, but it also highlights required and missing metadata to make sure researchers can prepare their data for reuse following publication to the SPARC portal. So when should you use MicroFile Plus? We recommend you use it as you are preparing your dataset for submission to the SPARC curation process or to the SPARC portal eventually. The MBF SPARC curation team also uses it now and will continue to use it during the dataset curation process to make sure that the datasets on the portal include this viewable and reusable image data that meets the SPARC information standards. Since you are the experts of your data, though, using MicroFile Plus prior to dataset submission will help expedite that curation process and get your curated data onto the portal faster.
Yeah, MicroFile Plus can also be used as a resource within your lab outside of SPARC. By writing the metadata required for image interpretation directly to the image file, it's easy to archive microscopy image data that you may reuse in the future. This is additionally beneficial if you're attempting to share or disseminate your image data within or outside your lab. It is known that this metadata is important for interpreting images collected during an experiment, and we know this is widely recognized because many researchers attempt to indicate information like Z-step, channel labels, subject IDs into the file name itself. There are many different shorthand strategies used to cram this necessary information into the file name. While this can be helpful for those within your lab who know that shorthand, having experimental details stored within the structured metadata, defined and standardized fields within your file itself takes the guesswork out of interpreting that file name shorthand. These abbreviations can even cause confusion within the lab. I'm sure you've all experienced rotations of postdocs or undergraduate students who need to be brought up to speed on this shorthand.
Good point, Aiden. And in another case, it could be someone outside of your lab too, maybe another researcher or a tool builder working to create a new image analysis tool that doesn't have the same level of expertise about the data, but they still want to interpret that data without doing a lot of heavy lifting. In this case, there are benefits to storing that image metadata within the image file in a defined structure. I'd also like to point out that describing this at the study or data set level is not usually enough because a single experiment can utilize multiple imaging modalities or experimental parameters. Each sample can include different stains or target labels, image scaling, objectives, or taken from different devices. For this reason, the best suited location for this information is right there, right inside the image file, independent of everything else. Storing this metadata directly to the file using MicroFile Plus can ensure that the correct and complete set of metadata is linked with the unique image file by the person that knows the experiment best, which is you. The researcher who carries out the experiment and collects the image data is the expert on the details like these target labels or the stains used. They can store this information directly to the image with MicroFile Plus. And down the line, others will be able to interpret it better.
We understand the variability between microscope devices that are used by researchers, and we have integrated support for a variety of microscopy image formats into MicroFile Plus. Here you can see a list of the supported file formats from a variety of different hardware vendors like Nikon and Zeiss. File format support is a continual process, and each of these file types can have edge cases or variants that will need to be supported individually. If we do not currently support your file format, contact us on the MicroFile Plus web forum. The link's at the bottom of the page here. We appreciate when users are willing to collaborate and share example image data with us so we can continue to improve MicroFile Plus's versatility.
Absolutely. Using MicroFile Plus is quite easy and has several key features. Here you can see a quick video of a typical conversion workflow. Aside from the conversion of files to the OME TIFF and JPEG 2000 formats, MicroFile Plus has this metadata generation window for you to review, add, or amend the experimental metadata. When there are missing fields, the files will have a red X, and that application will not let you convert your source image data until all the required metadata has been added. This acts as a really nice check or validation point. In the metadata window, the SPARC standard required fields are highlighted in yellow. Again, visibility of what you need to add to your data in order to meet the standard. And once all the metadata has been edited, you just click convert and your FAIR image data is going to be saved to the directory that you indicate. So to start, there are 4 different types of conversions available. First, you can convert your source image data from a single 2D or 3D image to a single 2D or 3D image in the OME TIFF and JPEG 2000 format. This is the most basic type of conversion and one we use most frequently for curation. With this option, you can also create single images out of container files like LIF or CZI. So if you have a CZI that contains multiple scenes, you can use this option to output the individual scenes as individual images and add unique metadata to scenes if applicable. Secondly, you have the option to convert many 2D images into a 3D stack. This is great for when you have a bulky TIFF series or DICOM series and you want to put them together into a single file that's going to be easier to share and store. Third, you can combine single channel 2D or 3D images into a multi-channel image or stack. We see this a lot with Zeiss LSM files where each image contains information from only one channel. So merging these together makes the analysis easier and things helpful for how those different targets interact or co-localize. Lastly, images can be combined into minimum, maximum, or deep focus projections. This isn't used as part of the SPARC imaging standard, but could be used by your lab to, say, create max projections for publication figures, for instance. Next, you'll see options to choose your save format. So there are your output file format, JPEG 2000, OME TIFF, or both. And for the JPEG 2000, you can indicate a compression, lossy compression, or opt to do lossless. For OME TIFF, it will always save or convert as lossless compression. And here's a closer look at the metadata generation window. You can see experimental metadata fields here, such as target label, the pseudo color that would be the display color of that channel, the modality. So there's some options according to the OME TIFF specification to choose your modality of the instrument you used. And you'll see that when there is missing metadata, that the field is highlighted in red, which again alerts you that you need to add that information. And then the yellow field labels highlights are to indicate that those are required according to the SPARC standard. And the program also allows users to create experimental presets that can be used to fill in multiple metadata fields for a group of images or a single image that were derived under similar experimental conditions. It's a huge time saver and can make adding that required metadata to a large selection of source microscopy image data so much easier. And last, but not least, we don't need to flip the page, just saying, sorry. And last but not least, for all the supported file formats, MicroFile Plus preserves the existing metadata written to the source image files. So you don't need to re-enter that information. You just need to add your missing fields and convert.
Thanks, Macy, for showing us what MicroFile Plus is capable of. Now I want to take a look at the big picture and show you the ways that standardizing image formats will impact the datasets hosted on the SPARC portal. If your SPARC dataset includes the web streamable JPEG 2000 versions of your microscopy images, they will be displayed in an integrated image viewer in the Gallery tab of the dataset. Users can explore these images on the web at full resolution just by selecting the thumbnail. And then you can even explore 3D image data plane by plane, like the one you see on the screen right now. This video, in the video here, this data was generated by Dr. Puking Uen from the Dr. Tache lab at UCLA. These views give portal users an idea of what imagery can be found in the dataset before they download those resources.
Yeah, there's a variety of lovely image data available to peruse on the portal now, and I'm excited to see more as datasets continue to be published to the portal.
Yeah, we encourage you guys to check those out at just go to sparc.science.
Yeah. All right, I think that's all we have today. Please feel free to submit questions if you have them now, if you haven't already. I'll give you a second to think of those. In the meantime, here are some resources. First of all, you want to download MicroFile Plus. You can go to www.mbfbioscience.com/microfile plus, and you'll see instructions on how to download, install, and license the software. And if you need help with SPARC or using MicroFile Plus, please contact us. You can email us at sparc@mbfbioscience.com. You can contact us on Slack on the NIH SPARC channel or the hashtag MapCore-support channel within that Slack. Or create a post on our forum. We have a MicroFile Plus forum, or our MBF Bioscience has a forum, and there's a MicroFile Plus dedicated page, and the link is there on the bottom. So we look forward to hearing from you.
Thanks, Macy. It looks like we do have one question so far. I'll read that off, and maybe you can take that, Macy.
Okay.
The question is, if I have pictures of my experimental protocol of sample preparation, for example, do these images need to be converted and abide by the SPARC image standards?
No, they do not. Only what's considered the source microscopy data that you use for analysis. So not ancillary supporting images for documentation or things that you would normally put in a publication. Just the data products suitable for analysis are covered.
Thanks, Macy.
All right. Well, I think we can wrap up if no one else has any more questions. We'll save you all a few more minutes of your day. We really appreciate you coming and attending and listening to us talk about this, and please reach out if you have any questions.
Thank you, guys. Have a great day.